Can you guys see my screen?
Yes.
Okay, I'll open.
Yeah.
Okay, thank you. Yeah. So throughout the lecture, if you have any questions or anything, you can type in the chat, or yeah, you can unmute if you want. Okay, first of all, how are your preparations going for MOL?
Alhamdulillah.
I hope they're going good. I hope like these past sessions are helping you in your preparations too. But inshallah it will go okay. So today I'll talk about protein synthesis, or like an introduction to protein synthesis and how it's being made in our bodies. Since we know that we need proteins, like our cells make it's an essential component in our bodies. They're being made every day. They're used for many different functions in our body. And if you ever need to ask anything, this is my number and that's my email. So wait. So these are the objectives that are given to you, and I'll try to cover all of them in this hour. Okay, so before I begin, what do these have in common? Can anyone tell me? They're anything, it's fine. Yeah, that's true. But since I'm giving a lecture on protein synthesis.
Bodies.
Yeah. Okay, so yeah, they're cartoons, they're energy. Yeah. Okay, so they have, like, surprisingly, they have proteins named after them in our bodies. But I liked all your answers. They're really good. They're all correct. So yeah. So when I was making this lecture, I found that there was something called the Minion Protein, and I know you guys took Sonic Hedgehog protein in the first year. So yeah, those are proteins, and that's like how the process of them being made. I'll go through them step by step. Okay. So first, we have DNA in our bodies, and they have a code of bases, and they have like this specific sequences to make these proteins. So there are two important processes that happen for the code in the DNA to go into making proteins. So the first step is transcription. Second one is translation. Okay. So how many bases are there in the DNA? So our DNA is made up of how many bases? No, bases. Like how many types of bases do we have? We do have 20 So we do have 20 amino acids and 4 bases. So there's thymine, adenine, cytosine, and guanine. Okay, that's correct. And they a combination of 3 of them, for example, TAC here or CAC, they make something called codon. So these are the codes that are found in our bodies, and so we call them codon because each codon, it makes, like, it translates into 1 amino acid. So three out of the 4, if you have 3 bases, that's 1 codon, 1 amino acid. Okay, and then this whole process is so when it gets copied into something called mRNA, that's called transcription. Now mRNA is a direct copy of the DNA. It's the exact copy. It's just complementary. The only different thing is that there's uracil instead of thymine. Okay, and everything else stays the same. And then mRNA, sorry, okay, someone's asking what is 4. I mean 4 bases. So we have 4 bases that make up our DNA. So thymine, adenine, cytosine, and guanine. Okay, is that clear? Okay, so again, so the information in our DNA, okay, they come in codes. They go into mRNA. It's an exact copy but complementary and with uracil. By a process called transcription. Now, the reason we have mRNA is because the DNA can't leave the nucleus. It's always in the nucleus, right? So the mRNA takes this message and it goes to the something called the ribosomes, where it can start the protein synthesis. Now mRNA or messenger RNA, it goes to ribosomes and it starts translation. So it translates this code it has into something protein, something called proteins. And proteins are basically chains of amino acids. And I'll go through the synthesis of this. Okay. Now, wait, so this table was given to you, and these, so like I said, three of the bases together, they make one codon or one amino acid. There are some properties of genetic, like this code you need to know. So the first one is that this code is unambiguous, which means it's specific. So let's say like so there are different combinations, right? So UUU codes for phenylalanine, for example. So you guys, do you guys know the amino acids in our body? Like you had a lecture on them, right?
Yes.
Okay. So yeah. Okay. So that's what I mean by being specific. So each one codes for a specific amino acid. Now they're also universal, which means all of these codes, they're all the same. Like they code for UUU codes for phenylalanine in all of the cells, in all the cells in our body. And then there's the third property is that the genetic code is degenerate, or it's sometimes called redundant. And I'll try to explain what that means. So for example, let's say in this one, CAU and CAC, even though they're different combination, like the last letter, if you notice, is different, they still code for histidine, which is an amino acid, sorry. So CAU and CAC codes for the same amino acid. So that's what degenerate means, that the third base, it doesn't really matter, it's fine. It can still code for the same one. So these are the three properties. Again, specific, they're universal, they're the same in all of our cells, and they're degenerate. Now, so the number of combinations we have of these bases are 64. Why? Because we have 4 bases in our bodies. Sorry, so someone's asking me to revise them. What do you want me to revise? Oh, the amino acids, you mean? Okay, so I won't go over the amino acids, but I can go over them briefly because this is just an introduction to it being like the proteins being made from the amino acids. So yeah, I'll go over them. Universal means it's the same in all organisms. Yes, yeah, you're right. Okay. So again, like I said, there are 64 different combinations of the bases. Now, in total, we have 20 amino acids. Again, why? Because some of them are degenerate. So even if the last base is different, it can still code for the same amino acid. Now, the important thing you need to know from this is the start, like you don't need to memorize all of this. You don't need to memorize which one codes for which amino acid and so on. What you need to know is what's a start codon and what's the stop codons. So the start codon will always be AUG or methionine. It's the only one that starts the whole process. It's always at the beginning of that amino acid chain. Okay, so AUG is methionine or the start codon. Now the stop codons, on the other hand, there's 3, and they sound the same. They're all like U, G, and A. There's just a different combination of all that. So UAA, UAG, and UGA. They're all stop codons. So they don't code for an amino acid. Whenever the ribosome sees these, they just stop like making the protein. Okay, they just cut it short. Is that clear for everyone? Okay, so again, so start codon and then stop codons. That's all you need to know from this table. And also, I just want to clarify something. So this table is based on mRNA. So that's why you'll see U over here, okay? The uracil instead of the thymine. Okay, that's all. So since I explained to you what codons are and so on, so 3 of them again make a code or a codon for one amino acid. Now there can be like sometimes there can be errors or mutations that can happen in these bases. So let's say, so there are two types of mutations, two main ones. There's point mutation and there's something called frame shift mutations. So point mutation, from the name, it's when one of the bases, they get, like, there's an error when placing that in the mRNA. So for example, if we take the codon we take the base UCA, it codes for a protein called serine. Okay. Now let's say the first base got changed into C, cytosine, and this completely changes the amino acid. So it's now coding for proline, something else. So these names that I'm saying, they're all amino acids. Okay, so when you have a mutation that changes the amino acid, that's called missense mutation. So you can memorize this by how I memorized this was like mistake, mishap, you know. So there's missense mutation, the amino acid that is coded gets changed. Now let's say there's a mutation, one of the bases got changed, and there it changed the code to a termination codon. So when we go back here, there's UAA is a stop codon. So here, when it gets changed from a normal amino acid to a termination codon, this is a nonsense mutation. Okay, so there's missense when it gets changed to a different amino acid, nonsense when it gets changed to a stop codon. Now the third one is silent mutation. So from the name, you can tell that nothing really showed up on the amino acid expression. So let's say this one, the last letter got changed to U. It will still code for the same amino acid because, like I said, the bases, like the codes, they're degenerate, which means that the last base doesn't really matter sometimes. It's okay to have it different and still code for the same one. So this is silent mutation. Are these three clear for everyone? Because this is like mainly asked in the exam. Okay. 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 And then, so there's missense, nonsense, and silent. The next one is frame shift mutations. And from the name, you can tell that the whole code or the whole sequence gets shifted kind of because of an addition or deletion of a base. So this mainly happens when you add one or two bases to the sequence, or you remove one or two bases from the sequence. So here you'll see an example where a random base is added, and then here where there's a deletion of a base. Now this changes the whole sequence of the amino acids that are being produced, and that's why there's a frame shift mutation. Okay, it changes the whole structure of the protein because the order is different, you know. So frame shift mutations are more like dangerous than, or it's more severe in a way. It changes a lot of things than the point mutations. Now, what kind of mutation is this? So this is the normal one, and this is the abnormal one. Yeah, it's, wait, I'll wait for others. Yeah, it's E. Okay, so and I know what I said about addition and deletion, okay, but in this case, there's three bases that are removed. So phenylalanine is removed from this whole sequence, but the rest of the sequence here, glycine, valine, and serine, they all stay the same. It's just three of the bases got removed. So in this case, we'll call it deletion because it didn't affect the sequence at all. If it did affect, we would call it frame shift mutation. So yeah, the answer is D. Also, I have the answers written in the notes if you guys need the PowerPoints later on. Okay. Now moving on from point mutations and frame shift mutations, we'll go on to diseases and how it affects our body, because that's what you need to know. That's what a clinical scenario can ask. Okay. So the first one we have is sickle cell anemia. And sickle cell anemia, whenever they ask you, it's a missense mutation. But the thing you need to know is the position and what it's changed into. So this is the normal sequence. You have glutamic acid. So someone wanted me to revise on the amino acids. Basically, you have 20 amino acids, and all of these, like valine, histidine, leucine, threonine, proline, it's yeah. So you'll have a lecture on that. You'll have another PAL session on that actually. So this one is for glutamic acid, and there are other amino acids too. Basically, glutamic acid gets changed into something called valine. Okay, here. So you can see the structures here. They're completely different. Glutamic acid, from the name, it's acidic. It has an acidic side chain, while valine is non-polar. It doesn't have any charge. It's not acidic, not basic. So this only one change in amino acid changes the whole structure, and it causes a disease as severe as sickle cell anemia. So what you need to know from this slide, in sickle cell anemia, glutamic acid, it's one of the bases get changed, so missense mutation, and it gets changed to another amino acid, valine. Okay, at position number 6. Is valine hydrophobic? Yeah, it is. So yeah, apart from the amino acid names, you have to know the properties of them too in that lecture. Okay. Again, okay. So did everyone get that? So glutamic acid gets changed because this is really important for the exam. The glutamic acid gets changed into valine, position number 6, and it's a missense mutation. Okay. Now, in sickle cell anemia, what happens is so you have the protein that gets misfolded and everything. It's abnormal. So the hemoglobin, it kind of like changes its structure in the RBCs. Okay, wait, I'll re-explain just a second. So the hemoglobin, it changes its structure in the RBC. That's why you have RBCs that are sickle shaped, like this in picture number 2. And that's why they can clump in capillaries, they can, stop blood flow, and you get all these symptoms. Again, you don't need to know all of it. You don't need to know that. But in clinical scenarios, you have to know that whenever they give you, for example, anemia, someone who came tired with severe pain, with vision problems, okay, you have to think of sickle cell anemia because the next question might be on mutation or what kind of mutation is it. Okay, so that's sickle cell anemia. Now, someone wants me to re-explain frame shift, so I'll briefly go over that, okay? So frame shift mutations. So can you tell me if you got point mutations? So point mutations is when there's one change in one base, change in it, okay? Frame shift mutations is when you have new bases added. So you have either 1 or 2 bases added or 1 or 2 bases deleted from the whole sequence. So this deletion or addition, it shifts the sequence kind of, and every 3 reads for an amino acid that's different from the original sequence. Did you get that? Or so you see the proteins, the amino acids here. So point is change and frame shift. Yeah, exactly. Yeah. So point mutation, you still have the bases. It just got changed into something else. Okay. Okay. What's the difference between frame shift? Do you get that, Leanne? Okay. So point shift again. Sorry, point mutation, the bases get changed. Nothing gets added, removed. But when you have addition and deletion, it changes the sequence. Like for example, here you have amino acids here, right? But when you add something, everything gets shifted and everything reads for something else. Now, the example I was showing you guys here, this one, this is when three of the bases got deleted. 3 here, T, it goes for phenylalanine, it gets deleted. The rest of the amino acids, they're still the same. Nothing is affected, okay? That's why it's a deletion here. The whole mutation here is deletion. Do you get that? Because it didn't affect the rest of the sequence. When one of the wait, so how is point mutation different to deletion? So deletion, what I mean by deletion is that a base or anything, it gets deleted. Yeah. So this does cause a disease. I'll explain it later, but it's cystic fibrosis. So it's an example, but yeah. Do you get that, or is that clear? So deletion is a different kind, you know? Yeah, okay. So what I mean by deletion is it gets deleted. That's it. That's all I mean. Addition is addition, like addition of the things. But these deletion, addition, it can fall under point mutation or frame shift. Frame shift is when it shifts the sequences. Point mutation, it's when it's like this. So deletion or addition, okay, can, in some cases, it doesn't cause frame shift mutations. Why? Because 3 of the bases, they get removed or added. So it doesn't affect anything else because 3 is the code, you know? Did you get that? Did everyone get that? Because if you still want me to explain, okay, I don't mind. Okay, I'll move on. But if you have any questions, yeah, if you have any questions, let me know. Okay, so deletion and frame shift are both under point mutation. No. So point mutation is something else. Frame shift mutation is something else. Okay, point mutation, only one base is changed, but it doesn't affect any of the other sequences. Frame shift mutation is when something gets added or yeah. So what I mean by that, yeah. So frame shift affects the whole sequence. That's the only difference between frame shift and point mutation. Deletion can happen, it can happen on its own. So here, it happened on its own without frame shift mutation, or it can have frame shift mutation. You get that? Do you get that? Yeah, okay, good. So if you're still confused by the end, just let me know. I have questions too at the end. Okay. Okay. So like I said, sickle cell anemia. Does anyone remember what kind of mutation is sickle cell anemia? Because I just went over it right now. Okay, yeah, missense. Why? Because glutamic acid gets changed into valine. It's a different amino acid. Now, wait, so yeah, so you asked me, right? So in this case, only one base got changed and missense. So under point mutation, we have missense, we have nonsense, we have silent. These are all point mutations. These are different types of point mutations. And that's completely separate from, yeah, exactly. Point mutation, missense. Now, the other disease I want to go over is Huntington disease. By the way, these are just examples to show how this can affect, but they can get these questions because they taught you in the lecture, right? So Huntington disease is when proteins, they become abnormal, they clump together because they're not the right shape, and our cells destroy them because they're not, they're not the normal forms in our bodies. So when they get destroyed, the neurons in our brains get destroyed, and that's why you see the brain kind of shrink in this picture. So that's Huntington's disease. It has cognitive decline. It affects a lot of functions in our body, and the protein that's affected is Huntington, okay, from the name. So the main problem here, okay, it's not point mutation, it's not missense, it's not frame shift. The only problem here is that there's a code CAG, okay, there's a code that repeats a lot of times, a lot more than it should be. So CAG gets repeated a lot of times here. Can you see that? So here the protein is not folded in the right way because it's too much, and then that's why they kind of like clump together, and then the neurons get destroyed. Does that make sense? So we talked about sickle cell anemia. It was a missense mutation. Now we're talking about Huntington disease. It's a trinucleotide repeat expansion. This code is repeated too many times, and it disturbs the whole protein. So they can ask in the question, they can get you. I remember we had CA, so we needed to know that CAG gets repeated a lot. Okay. So I'll move on. If you have any questions, let me know. Now, the third one is cystic fibrosis. Now, again, cystic fibrosis is an example of a deletion mutation. Okay, So it doesn't cause, it's not a point shift because it's not one base. It's not frame shift because it doesn't shift the, it doesn't shift the sequence. It doesn't change the amino acid that come after it. So cystic fibrosis, what it means, what the protein that gets affected is a channel we have in our like respiratory tract. It's like CFTR protein or channel. And its normal function is to pump chloride ions out of the cells so that the mucus can stay hydrated, so that water follows it and mucus stays hydrated and so on. But whenever it's abnormal, whenever that deletion happens, the chloride ions don't go out of the cell and the water stays inside the cell, and that's why the mucus kind of like dries up and it traps a lot of bacteria and you have lots of infection and so on in cystic fibrosis. So the mutation, so this is the question that I showed you before. So phenylalanine, which is coded by TTT, 3 bases, okay? It gets, the whole thing just gets deleted. It doesn't cause a frame shift mutation. It doesn't shift, so you get what I mean, right? I think you might be confused because I'm saying shift. It does shift, but like the amino acids here, they don't get changed. They're still the same. Glycine is here, glycine is here. Valine, valine, serine, serine. Okay? Okay. I'm explaining just so that everyone knows. I'm not confusing everyone. Okay. So that's it. So that's a deletion and the position here is important. I'll answer that. So the position here is important because they can ask you this again. So position F, I memorized it by phenylalanine, so F, and then 508 is the position, it's the position of the amino acid. That's it, you just memorize it. So F508 is where when there's a deletion of phenylalanine, you get cystic fibrosis. Like you can see how a small change or a small deletion can cause a lot of problems in. Okay, so someone's asking, so it will keep the water inside the cells, yes. So when chloride ions cannot be pumped out, water stays inside too, it doesn't go out. So the mucus kind of like dries up. Does that make sense? So the mucus kind of gets sticky, more sticky. That's why it causes all the problems. Okay. Do you guys have any questions so far? By the way, this is my cat.
Great.
No. Okay, if you guys don't have any questions, I'll move on to the next part. Thank you. Okay. Oh. Okay. Now the next part of this lecture.
Did you tell them the name of the class?
Yeah, so his name is Tomo-chan. Okay, I'll just move on because there's no questions.
Momo is Japanese.
No.
Do they want to play?
Oh, do you guys want a break, by the way? Because just to pray? Because I can stop and I can start the translation part after this. Okay. I'll continue. Okay. Okay, I'll continue. Okay, so there was another lecture on transcription. So transcription, yeah, so there's 2 processes, like I said, transcription and translation. Transcription is when DNA becomes mRNA. Okay, it creates the messenger RNA. Now for this next step, what I'm going to explain is translation. It's when the messenger RNA goes from the nucleus, goes into the cytoplasm, goes on something called ribosome, and this will start the protein synthesis. So proteins, as you know, they're a chain of amino acids. Their building blocks are amino acids. And the thing you need to know from this lecture is what you need for the protein to be made. Okay, so you have amino acids, obviously, because they're the building blocks. You have the mRNA, which carried the code from the DNA. And then the next thing is there's something called tRNA or transfer RNA. Okay, so these 3, they go into an organelle called ribosomes, and the ribosome kind of like takes all these components and they make protein. So ribosomes are the site of protein synthesis. That's what you need to know. And they need all these components. Okay. So before I move on to that, I want you to know the different types of RNA in case anyone's confused, in case you get confused in the exam. So there's mRNA, rRNA, tRNA. Messenger RNA is messenger RNA, like I said, it's a copy of the DNA. That's it. It takes the information. rRNA, so messenger RNA takes the information to ribosomes. Ribosomes are made up of an RNA called rRNA, ribosomal RNA from the name. tRNA or transfer RNA gets amino acids. They have amino acids on each of them. So here, for example, transfer RNA, they have an amino acid. They bring it to the ribosome, they read the messenger RNA, and the amino acids kind of build up together to form a protein. That's it. So tRNA, rRNA, mRNA. Now, again, these are all of the things you need for protein synthesis to occur. So please don't forget them. And the site is, again, ribosomes, which is in the cytoplasm. First component I'll go over is tRNA or transfer RNA. Now I'll go over the structure real quick. So imagine this structure was straightened, it would be the same thing as mRNA, okay? But it's in the opposite. So you know how everything is 5' to 3', that structure, right? So tRNA, it's the exact same. It has a 5' end, it has a 3' end. The only reason it's like this, it's just folded by hydrogen bonds in between here, okay? So it's just folded like that. And two important things they can ask you in the exam is the amino acid, like the amino acid, where is it bound to in the tRNA? So it's always bound to the 3' end. So you'll always find the amino acid on the 3' end of the tRNA. Now, the other thing I want you to know is the anticodon. So they have something, at the end of its structure, they have something called anticodons. Now remember codons from the DNA and onto the mRNA, they're like the three bases that code for an amino acid. So this is the exact thing. It's called anticodon. The only difference is it's complementary. So it's the opposite of the mRNA, but it goes from 3' end to 5' end. That's it. So the mRNA here, it's read in the direction of 5' to 3'. So the tRNA kind of like reads from 5' to 3', the mRNA, and the amino acid that's at the end of it, it joins together to form the protein. I'll go over that. Okay, 1 second. So it's read again 5' to 3'. Now, before I move on, that's tRNA and mRNA. They kind of go together, they read it, that's it. So the codon in the mRNA gets bound to the anticodon in tRNA. Now, there's something called the wobble hypothesis. So remember how I said the DNA, the code in DNA is degenerate, which means that the third base, even if it's different sometimes, it can code for the same amino acid, okay? The same is true for this. So you know how the normal pairing is between G and C. It's always C with G together, okay? But in some cases, there can be U, for example, with G. There can be A with G, but it's okay because the last base here, it's okay for it not to be the same always. So that's called the wobble hypothesis. That's why you have a lot more like tRNA, than there should be, because two of them could be for the same mRNA code. Does that make sense? Like wobble hypothesis? Because I remember when I was confused in first year with this. So the main idea is that the third pairing, it's okay for it not to be the same. That's the whole idea of wobble hypothesis. Okay, so yeah, it's pretty clear. That's good. Does it cause disease? Someone's asking. No, it doesn't cause disease. That's okay. It's a normal phenomenon. It's okay for it. I don't understand if the tRNA starts from 3 or 5. Okay, I'll go with 5. Okay, so do you get that the mRNA is from 5' to 3'? Remember, it got copied from the DNA. Okay, so that's 5' to 3' of mRNA. Okay. Now, for the anticodon to match onto this mRNA sequence, it has to be complementary. So it has to be the opposite strand. So it has to be from 3' to 5'. The only reason you're not seeing it in a straight line here, it's just folded. So 3' is on this end, okay, and 5' is on the other end. And it just matches the 5' to 3' of the mRNA. The only reason it's opposite is because it has to be, you know, opposite of the mRNA. Did you get that? It's not, you don't have to think too much about it. Like, if you didn't get it, tell me, I'll try to re-explain, don't worry. Okay, you got it? Okay, so again, it's just, even in the DNA, it's like this 5' to 3', and then the opposite strand is 3' to 5'. It's just the opposite. Okay, now again, so I explained about tRNA and the wobble hypothesis. About the third, you know, the third pairing, it doesn't have to be the same. Now the second thing you need for translation, the first one, tRNA. Second one, mRNA, messenger RNA. You saw this everywhere in DNA replication too, but mRNA synthesis. Okay, so mRNA comes in, this purple thing here is the mRNA. It goes through these structures, these blue structures are ribosomes. It goes through it, and the protein is made out of the ribosomes. Now, when you have a lot of these ribosomes making it at the same time, you have a lot, right, here. So this is something called polysome. It's just a name for a lot of ribosomes doing the same thing, making the same protein. Okay. Now, again, the third thing is ribosomes. So you have your tRNA with the amino acid, it's ready. You have your mRNA with the code, it's okay. Now what brings them together is the ribosome. The ribosomes, so I'll try to explain this because mostly this is mostly based on memorization. I remember I used to remember all of these like subunits, but I'll try to go over them so you guys can memorize them together with me. So you have prokaryotes and eukaryotes. Eukaryotes have a nucleus, prokaryotes don't, they're in the bacteria and so on. Okay, so what matters is in eukaryotes, you have a different kind of ribosome than a prokaryote. In eukaryote, you'll have an ATS ribosome. It's a bigger ribosome. So S just means it's a unit they came up with. So when you spin down the ribosomes, it depends on how fast they fall. So they just named it S. So ATS. Just know that ATS is in eukaryotes, 7TS is in prokaryotes. ATS in eukaryotes is a larger ribosome. So it has two subunits. Both of them have two subunits, a large subunit, small subunit. You can see in the picture here. So large subunit is 60S, small subunit 40S. 60 and 40, they join together, the whole thing is called ATS. In prokaryotes, however, you have the large subunit is 50S, small subunit is 30S, and the last, like the whole ribosome is called 70S. It's just memorization again, but I know you took it in high school too. Like I remember taking ATS in eukaryotes, 70S prokaryotes. Okay, the thing you have to memorize from this, like new thing, is these. So the large subunit, remember how I said the ribosomes are made up of protein and rRNA. Okay, so the RNA that makes up the ribosome is rRNA. It has its own components. So the large one, so let's start with prokaryotes, for example. We have the large subunit 50S. It's made up of something called 5S RNA. It's the size, okay, 5S, 20S RNA. For me, I don't think you need to remember the proteins because they'll only ask you, like, for example, if they ask you where 23S is, it's in the 50S subunit of the prokaryote. Okay, so it's just memorization at this point. So 30S, the small subunit of this ribosome, has 16S RNA. In eukaryotes, again, they're made up of rRNA, but they're different. So 5S is the same as prokaryotes. 28S is close, it's bigger in eukaryotes, so you can memorize 28 and 23 in the prokaryotes. And then there's an extra one because it's big, so 5.8S. And then the small subunit has one. It's kind of close, 16 and 18, but 18 because it's bigger, because eukaryotes are, bigger. They have a nucleus. Is that clear? It's all memorization. But if you have any questions, you can stop me. So again, ribosomes, they're made up of rRNA, and I just tried to tell you what they're made up of before going into how they're being used to make proteins. Now again, so we have small subunit. and we have large subunit, regardless of whether it's ATS or 70S. Now we have three sites, three important sites in the ribosome. We have A site, P site, E site. So remember how I said tRNA and mRNA comes into this and they make proteins, right? So mRNA, it binds to the small subunit first. So it binds here, okay? Here. mRNA comes in here and the ribosome kind of reads it like this. It goes like that. And then the place for tRNA are these sites, A, P, and E. So tRNA comes here and then comes on the P and then exits from the E site. I'll try to go over them, okay? So these are the steps. I'll go over them. I just have one more point about this one. So the three steps in translation you need to know: initiation, elongation, and termination. They're easy to memorize. Initiation, start; elongation, you elongate the chain of amino acids; termination, you end the process. And then you get your protein. That's it. So before I begin initiation, there's so I know you guys took this lecture and there was something called the Shine-Dalgarno sequence at the beginning. So this is only for prokaryotes, like it says here. It's a prokaryotic mRNA. Sorry, from 5'? What do you mean? From 5' here?
Can you go to the previous slide?
Yeah, sure. This one?
Yeah. Is this start reading from 5' to 3'?
Mm-hmm. Okay, so yes, you read the mRNA 5' to 3'. So 5. Can you see my mouse, by the way, here?
Yeah. Yes.
Yeah. Okay. So 5'. So the mRNA is here. Okay, it's the 5' end, and then the 3' end is all the way at the end. So the 5' goes in like this, and the ribosome.
Okay.
Read the mRNA in that way. Okay.
Okay.
Sorry, can I just ask a question?
Yeah, sure.
So you know how tRNA is 3' to 5' because mRNA is 5' to 3'.
That's
So
correct.
how is it synthesized? Is it synthesized the same 5' to 3'?
Okay, yeah. You know how in DNA you made the mRNA, right? But in this case, you're not making anything. You're not making anything from 5' to 3'. Like it ends here, you know, that 5' to 3' thing. Because listen, wait, I'll go over the steps. But basically, mRNA comes here, 5', and that's 3'. tRNA is from 3' to 5', so just so it can bind to the codons on the mRNA. Okay, 3' to 5'. But at the end of all this, you're making a protein. So you're making amino acids bind together, they make a protein. You don't have 5' to 3' of proteins. You get that? So what I said about tRNA being from 5' to 3' here, it's just to like make you understand that it's complementary to the 5' to 3'. But at the end, you're not making it. Just know that mRNA.
So there's no synthesis. It's just
No.
making amino acids.
No. Yes, exactly.
Okay, thank you.
Just know that mRNA is read from 5' to 3' because the ribosome kind of goes along it. So the ribosome reads this part and then it goes here. So this direction is 5' to 3'. Okay? Is that okay?
Yes, thank you.
Okay, no problem. Okay, so again, 3 steps: initiation, elongation, termination. But again, for prokaryotes, there's another sequence. It's an extra sequence for prokaryotes because, you know, there's prokaryotes. They're different from us. So they have something called the Shine-Dalgarno sequence at the beginning of the mRNA. And what you need to memorize is this part here. So this binds onto the 16S rRNA of the small subunit. I'll go back to the picture so you guys understand. So this is basically a sequence. Okay, it just helps in the mRNA being read by the ribosome. So mRNA is in the cytoplasm, right? It came out of the nucleus. It's looking for the ribosome. The ribosome needs something to know that this is mRNA, and how it knows that is by the sequence in prokaryotes. So here, it binds to the small subunit of prokaryotes because it's a prokaryotic mRNA, and to 16S. 16S is the only RNA in the 30S. So that's easy to memorize, right? So if you know that it binds to this one in this subunit, you're good. So okay. So this sequence for eukaryotics, it's transcription factors. Okay, wait, yeah, I'll come to the eukaryotes. So this one again, it binds to the 16S of the 30S subunit. That's it. So when it binds there, the ribosomes know that, oh, this is mRNA. And then it starts combining with the large ribosomal subunit, and it starts reading the mRNA. Like you can see here, this is AUG again, methionine. It's the start codon of the whole protein synthesis. Okay, for eukaryotes, so transcription factors are something else. They're in the DNA. They basically regulate expression of something. They're like proteins. It's something else. It's a different topic completely. But for eukaryotes, you don't have, the main idea is that you don't have Shine-Dalgarno. You just have, you have a sequence, it binds to the small subunit. Is that okay? Okay. So if it's, yeah, just let me know if that's clear and I'll move on.
Yeah. Can you repeat this slide, please?
This slide? Okay.
Yeah.
I will. So in prokaryotes, so this is mainly for prokaryotes. Eukaryotes, they don't have a specific sequence. They just have like a sequence that the ribosome can identify. But there's a specific one in prokaryotes. That's why I'm stressing on it. It's called, what sequence is it? No. There's no name for it. It's just a sequence. You don't need to know it for eukaryotes. So for prokaryotes, okay, yeah. I'll go back to that. Yeah. So again, prokaryotes. Let's focus on prokaryotes first. Okay. Prokaryotes have this specific sequence. Like it's a different sequence from anything else, right? It's called Shine-Dalgarno sequence. Okay. And it kind of like tells the ribosome that this is an mRNA, so that the ribosome can bind to it and start protein synthesis. So this part here, this sequence, helps the whole mRNA to bind to this one here, to this subunit, 30S. So because we're focusing on prokaryotes, right? So it binds to a 16S RNA on the 30S subunit. Is that clear, Rima?
Yes.
Okay, so that's the whole point.
So is it a promoter?
So promoter is something else on the DNA again. So promoter, wait, where did you guys take that? Which lecture? Do you remember?
No.
Okay. So I think you took that in gene expression, because what I remember is in the DNA, you have promoters, you have transcription factors. Promoters are a sequence in the DNA. It's not in the mRNA, what I'm explaining right now. So promoters, when transcription factors, they're proteins, they bind to these promoters or sequence in the DNA. It starts expressing that gene. Did you get me?
Yeah.
Okay.
It allows the RNA polymerase to bind in the DNA. Am I right?
Yeah.
Okay.
So it starts that process. That's in transcription then, yeah.
Yes. Okay. So
Yeah.
so Shine. Oh wait.
Yeah.
What is the
Yeah, I
name?
know. Shine-Dalgarno.
Dalgarno. Yeah.
Yeah.
Is it responsible to start the translation here?
So yeah, this is translation. The whole process I'm trying to explain is translation. Okay. But the amino acids, they don't start here. The initiation codon is right here. This sequence is just for the ribosome to know that this is an mRNA, and it just binds and then it goes along and reads it. Do you?
Oh, okay. Okay. So it's a bind site.
Yeah, exactly. Yeah,
Okay.
it's a binding site. I know it's hard to memorize the name, but yeah. Do you know the shrines? I just memorize it by that. Okay, someone was asking, yeah, so 5' UTR is also something else. It's in eukaryotes, if I'm not wrong. Yeah, 5' UTR.
Yes, it is, but I was asking
Yeah.
what's the function of it? Like, why is it there? What does it
It
do?
doesn't, yeah, it doesn't code for anything else. It's just,
Is
it's
it
just
for
a signal.
protection or?
Yeah, so I think what you're talking about is transcription.
Yeah.
Yeah. Yeah. So transcription is something else. Transcription is when DNA gets copied or made into mRNA. Okay. And in that process, there is 5' UTR in the mRNA. There is. And the main function, if you're asking for function, it's for protection because it has to go out of the cytoplasm. It is also for it's a signal, like to say that this is a ribosome, like this is an mRNA. Did you get that? So there's something also called capping in transcription. That's
Yes,
another
facts.
lecture. But yeah, exactly. Okay. Is everyone clear on that? Did I confuse anyone? Or
Clear.
okay, I'll move on. So initiation again. So this whole sequence that I'm trying to tell you is that it starts the initiation step because it binds and then it goes and reads the mRNA. Now, initiation codon, again, like I said, it's AUG. It's always AUG. It's always methionine. It's the first amino acid that starts the protein. Now, all of these, now this is for eukaryotes because regardless of it being eukaryote or prokaryote, the mRNA will bind to the small subunit. So the small subunit in eukaryotes was 40S. Okay, here. 40S is the small subunit of eukaryotes. So the mRNA comes here, it binds to the 18S RNA of 40S, and then the process starts. So the large subunit comes in here, tRNA comes, and then amino acids join together after that. Okay, so there you go. It binds to 40S ribosomal subunit because it's the small one. It starts small and then it goes big. Yeah. So now I'll explain the process of translation. And it's important not to confuse that with transcription because transcription is something else. Translation, it kind of makes the protein from that code in mRNA. So let's start with initiation. So initiation is when when mRNA binds to the small subunit, it moves along. Okay, it sees AUG in the code. That's the start codon. And then a tRNA for the complementary to the AUG comes in. It's the only one that goes directly into P site, the middle of the two sites. Okay, the middle of the three sites. So the tRNA goes into the middle, and it has methionine in the beginning. So let's say this one is, ignore this because this is so it has methionine. Okay, because methionine is AUG, it's specific. Okay, now that's the only one because it's the start codon. There's nothing before it. It's the only tRNA to go into the middle. Then the next codon here is read by the ribosome, like by another tRNA that's specific for it and complementary to it. It goes into the A site. So A site is kind of like the entry into the ribosome. Okay. Now it has an amino acid that's specific to this codon, that's specific to this code. So you have now two amino acids next to each other. Okay. Since you have two, since they're close together, there's an enzyme called Peptidil Transferase. You have to memorize that name, but it's an enzyme on the 23S rRNA that makes the peptide bonds between the amino acids. So the amino acids that's on the P site, it jumps onto the A site. So it leaves the P site here. Okay, if you can see, it leaves it empty. There's nothing else. So the chain continues on the tRNA here. Now these two tRNA, they move one space up. So the one at P goes to E, the one at A goes in the middle. So when it does that, E is the exit site. So the one that has nothing on top, it just leaves, while the one that has the chain of amino acid, it stays in the middle. Okay, and on the P side. And then this process keeps on happening. why is it 23S RNA? Oh, sorry, I'll go back. So 23. Oh yeah, you're right. Okay. So 23S RNA. So I think, sorry, if I said it's on the 23S, I mean it's made up of the 23S RNA, like ribosomal RNA, this enzyme. Is that okay? So yeah, I'm sorry if I confused you. So eukaryotes don't have the 23S RNA. Okay, yeah. The way they made these slides are confusing because they went here, look, they went from prokaryotes and then they talked about eukaryotes here. So yeah. So what do you need? Okay, I'll try to explain. Okay. So these are, yeah, this part is all memorization. So yeah, there's nothing else you can. Okay, so okay, listen, wait. Let me go back to the question. Okay, so the RN again 70S is the ribosome, the whole ribosome in the prokaryote. 80S is in eukaryotes. It's for eukaryote, everything's bigger. That's how I memorize. So 80S, for both of them, there's large subunit, small subunit. 50S, 30S for prokaryotes, 60 and 40 for eukaryotes. The way I did it is like 50 and 30 and 70, they're all odd number, like you know how 5, 7, and 3 are odd, and then here it's 6, 8, and 4, they're even, and eukaryotes starts with an E. That's how I did it. So does that help? I hope it helps. But anyways, so 60, 40, and 80, they're all part of eukaryotic ribosome. Now, all of these subunits, they're made up of rRNA, ribosomal RNA. Okay, and they're different for each. You can memorize them. To make it easier, you can know that 5S is found in both, and while the rest of them, they're just bigger than the prokaryotes. Eukaryote is bigger, so 28S, here 23S, here 18S, here 16S. Okay, and sorry, for someone, someone said they're confused about the 23S. 23S ribosomal RNA, it's just the size of the RNA. I'm sorry if I said it's for the process that I showed you here is for eukaryotes, but the enzyme that helps in it, the enzyme that helps in it is a ribosome, and it's made up of 23S srRNA, 23S ribosomal RNA, and that's why it's called a ribozyme, because it's an enzyme in the form of a ribosome. If that makes sense, is that clear? I hope that's clear. If not, I'll explain it again. Just let me know. Okay, okay. Which part do you want me to repeat? So this is for eukaryotes. The whole process I'm explaining. So the steps, they're the same for eukaryotes and prokaryotes. But I want to focus on eukaryotes. So okay, 23S part. This one. Okay. So the amino acids here, they join together by something called peptide bonds. Okay, so that's just the name between the amino acids. The way it does this is by an enzyme called Peptidil Transferase. Now, Peptidil Transferase, it's an enzyme, but a ribosome at the same time. So that's why they like combine the name. They're calling it ribozyme. This ribozyme is made up of 23S rRNA. That's it. That's why it's here. Now, I think like why we got confused is because 23S RNA is also found in this subunit in prokaryotes. Okay, so that's why. Is that okay? So ribosomes, all ribosomes, they're made up of rRNA, ribosomal RNA. And the numbers that I'm telling you right now, like 23S, 16S, they're the size, they're kind of a unit for the rRNA. So the one here is 23S rRNA. Is that okay with everyone? You can ask me if I can explain it again. Okay, I hope that's clear. I hope the numbers are clear too in the subunits. Okay. So this whole part that I'm telling you, this was a whole summary of the steps, by the way. Okay. So initiation is when, wait, let me go back. So initiation is when the start codon comes into the A, like into the A site, it moves directly onto the P because there's nothing else before it. It's the first amino acid that it carries. Okay. And then another one comes in at the A site, and then the amino acid shifts or it jumps to the other one, it joins by peptide bonds. That's how the whole process, it moves on like that continuously until it reaches the stop codon. So the initiation again is when the start codon comes in. Elongation is when this happens, when the amino acids, they join together to form a long chain of amino acids called the protein. This whole thing is the elongation. Okay. Here it says 23S rRNA of the 50S ribosomal subunit. This is again in prokaryotes. That's why it's kind of like they mix up the whole slides. But in prokaryotes, it's the 23S that carries out the peptide bond. That's the enzyme. Okay. Now, okay, let's move on. So whenever this process happens, whenever these two move on to this one, it's called translocation. And then you reach something called termination. Okay. Again, so termination, do you guys remember the code for termination? It's written on the slide, but so UAG, UAA, or UGA. These are the codons on the mRNA. Whenever the ribosome sees these, there's something called the release factor. So there's no tRNA for UAA or UAG or UGA. A release factor comes into the A site. Okay, it binds to something called GTP, it's an energy source. And then basically what happens is it allows the release of this chain of amino acids. It releases it out of the ribosome and everything goes away. And you have your protein. Is that okay? That's all. So initiation, start codon, elongation is when the amino acids join together, and termination is when a release factor. Okay. Yes, yeah. Basically, yeah. Because I saw that I think they mixed up prokaryotes with eukaryotes on this slide. So just know that. From my side, just know that. 28S in eukaryotes and 23S. Is that clear for everyone, this point? Because I know it can be confusing. Okay. So if anyone's confused, what that means is here, 23S is for the prokaryotic ribosome here. It's only found in prokaryotes anyways, so it's, yeah. And then eukaryotes, it's 28S. Is that okay?
So
Yeah. Sorry?
they wrote it wrong.
Wait, so I couldn't hear you. Sorry, again.
Did they write it wrong?
No, they didn't write it wrong. They just forgot to specify like prokaryotes, eukaryotes, or what that means, like in here.
So 23 stands for
23S, yeah, prokaryotes. 23S, prokaryotes. 28S, eukaryotes. Eukaryotes, everything is bigger. 28.
Okay, so the picture for prokaryotes.
Yeah. I just realized it now too.
Okay, thank you.
Okay, is that okay? I'm so sorry for mixing that up if I confuse anyone. Just know that 23S, that's why I told you to, like, it's important to memorize the rRNAs that make up prokaryotes, eukaryotes. So 23S rRNA is found in the large subunit of prokaryotes, 28S, the large subunit of eukaryotes. Okay. Okay. Now, remember the diagram I showed you with, so these three are the steps: initiation, elongation, termination. So there was a diagram I showed you before where there were a lot of ribosomes doing the same exact thing. So what's that called? Does anyone remember?
Polysome.
Yeah, polysome. Yeah. So they're called polysome or polyribosome because sometimes it just makes a lot of copies of the protein. That's all it is. That's all you need to know from this slide. Now you have this here. This one is a ribosome, right? And then this here, this chain is the protein that's made. Sometimes there needs to be changes done on the protein. Okay, there needs to be like things added, like phosphate groups, so that the protein can fold more, it can perform its function more. So in that case, it goes to different organelles. One of the main organelle it goes to is the endoplasmic reticulum here. Okay, endoplasmic reticulum is where most of the changes happen. So whenever it needs to go to the endoplasmic reticulum, it has a signal peptide. Again, for everything, you have a signal peptide. So there's something called the signal recognition particle. It binds to the signal peptide, and it kind of takes it, it recognizes that this has to go to the endoplasmic reticulum. So it kind of like takes the whole chain of amino acids and takes it to the endoplasmic reticulum. So that the protein can continue and it can be released into it. And then many changes happen after that. Okay, so sometimes it can even, so there are proteins needed in the nucleus itself. So in that case, you'll have something called the nuclear localization signal, NLS. So from the name, you can know that this signal will tell the, ribose or the protein mate to go to the nucleus. The signal for it to go to the endoplasmic reticulum is the N-terminal hydrophobic signal sequence. Is that okay? So it's just hydrophobic signal that goes to the endoplasmic reticulum. Okay, I'll repeat. So a lot of our like the protein, some of the proteins that's made by the ribosomes, they need to go outside of the cell. Okay, some of them need to go into the nucleus. Some of them need to go to the endoplasmic reticulum so that many changes can happen, and then it can package the protein in a nice way. It can release it from the endoplasmic reticulum. So for the cell to know where this protein will go, it needs a sequence. It needs a signal sequence, right? So I'll go back. So this one here, when the protein is being made, the first portion is a signal sequence. It has a specific signal for wherever it needs to go. So there's a signal sequence specific for the endoplasmic reticulum, again, where many of the changes happen. This signal is specific for the endoplasmic reticulum. That's why it's called N-terminal hydrophobic signal sequence. N-terminal, it's just, yeah. You don't need to worry about that because that means like the N-terminal of the amino acid. Okay, hydrophobic means it's not like non-polar, doesn't have a charge. When it needs to go to the nucleus, it has another signal sequence called nuclear localization signal. So this signal, it's specific for the nucleus, and that's why this protein gets directed to the nucleus. Is that okay? You don't need to know the specific sequence. That's why I'm not going into details. Is that okay?
So SRP is N-terminal hydrophobic signal sequence.
Which one? SRP?
Yes.
Oh, okay. So SRP, okay. So SRP is it's something else. It sees this signal peptide. It sees it. It attaches to it, and it drags it down to the organelle, so that it can release it can continue making the protein inside it, so that when it releases it, it's inside the organelle it needs to be in.
So
Okay.
SRP is only for ER.
Yes, yeah. there's something else for the nucleus. I won't go into detail because that's another lecture.
Okay.
Okay.
So what is the Interminant Hydrophobic?
Okay, so we're talking specifically about the endoplasmic reticulum. Okay. N-terminal. So you do you guys know the structure of an amino acid? It's okay if you don't. I'll explain it. But just do you have an idea about it? So an amino acid, it has an N, like one of its ends, it's the amide. So it has N at the end. That's what we call the N-terminal. And then the other end, it has carboxylic acid, so we call it the C-terminal. Okay.
Okay.
Okay, so you have N-terminal. So this signal sequence, it's just a name you have to memorize. It's the N-terminal of the amino acid right there, and
Mhm.
it's hydrophobic. It doesn't have a charge. That's it.
Okay. Thank you.
So yeah, no problem. But is that clear for everyone? Because if not, it's
Can I
fine.
ask the last question?
Yeah, sure.
So
Go ahead.
SRP for ER and NLS for the nucleus. Am I
Okay,
right?
so proteins, because yeah. So NLS, the nuclear, from the name nuclear, it's for
Mhm.
the nucleus. It goes to the nucleus.
Okay.
SRP, signal recognition particle, it recognizes the signal peptide. I know there's another name for the one for the ER, but this is just a general name, okay?
And are they the same, but the SRP for ER and NLS for nucleus? And they are
NLS is for,
doing the same
yeah.
thing.
So for example, like there's another specific name for it. I remember taking it in first year. There's another lecture for that. But that one is specific for the ER. Okay. And then the NLS takes it to the nucleus. It's something else.
Oh, okay.
Is that fine?
Okay, thank you.
No problem. Do you have any more questions? Because I don't mind. I'll try answering them. Okay, so I hope that's clear. So basically what you need to know is that there's a sequence, a specific signal for each organelle. If it needs to go outside of the cell, there's another signal for that, okay? And one thing I wanted to mention is that everything that goes through the ER, like the endoplasmic reticulum, it mostly goes out of the cell because it has other functions outside of the cell. That's why it has to get modified. A lot of things need to be added to it. So there's changes that happen to a protein after it's made. That's why we call it post-translational modification. Okay, and sometimes when a protein helps in the change, like the modification, we call them chaperone proteins because they kind of like help in the packaging of the proteins. The important thing out of all these is the changes, like the different kind of changes you need to know for like post-translational changes. So don't confuse that with post-transcriptional changes because that's something else again. Transcription is something from in the nucleus, from the DNA, it goes into mRNA. Post-transcription happens, I don't know if you remember, but there's something that happens to the tail end and the head end, there's capping, so that the mRNA is ready to go out of the nucleus. But post-translational change happens after the whole protein is made, like after the chain of amino acids is made, and it can happen, so I'll go through it. It can happen, mostly all these changes happen in the endoplasmic reticulum. The first one I want to go over is phosphorylation. So phosphorylation, from the name, it's basically when a phosphate group is added to the so phosphate group is added to an OH group of any amino acid, like specific amino acids. And these three amino acids are serine, threonine, and tyrosine. Okay. So serine, threonine, and tyrosine are amino acids that have an OH group. And phosphorylation can happen in them, which means that a phosphate group is added to them. because they can ask you where does phosphorylation occur and so on. So the way this happens is by an enzyme called kinase. It's a family of enzymes called protein kinases. And whenever you want to remove that, phosphatases come into play. So phosphatases, from the name, it breaks down that phosphate bond. Kinase is the opposite. They make it. They kind of make that bond. So this is phosphorylation, serine, threonine, and tyrosine. Okay. And then there's covalent. So the next modification is glycosylation. So so phosphorylation is when phosphate group is added. Glycosylation is when a carbohydrate group is added. So all these changes can happen after a protein is made. Okay. Now there's two types of glycosylation. There's O-linked and N-linked. So O-linked and N-linked, it's all based on the structure. It's all based on where it attaches to. So O-linked is when the carbohydrate group is attached to OH group of an amino acid, whereas N-linked is when it attaches to the amide nitrogen, or N, that's why it's N-linked, to of an amino acid. Now this carbohydrate can either be N-acetyl galactosamine or N-acetyl glucosamine, okay? But what matters is like this one here, it attaches to N, so we call it N-linked. This one attaches to O, so we call it O-linked. Now the ones that that undergoes O-linked are serine, threonine, and hydroxylation. Now serine and threonine, they're the same groups as before, right? They can undergo phosphorylation as well. So it's not that hard to remember. The other one is hydroxylation. So it's a lysine, like the amino acid here, you don't have an amino acid called hydroxylation, it's just lysine, and then there's a hydroxyl group attached to it. And that's why this carbohydrate can attach to that OH group, and that's why it's O-linked. Okay. For N-linked, it's the amino acid that can undergo N-linked is asparagine. And that's what you need to memorize. Is that clear?
Yes.
Okay, so again, for carbohydrates, you have O-linked attaching to O of serine, threonine, hydroxylation, because they all have the OH group. N-linked is when it attaches to the N, or the amide group, of asparagine. Now, the place where this happens is different. It's not only on the endoplasmic reticulum. So the O-glycosylation occurs in something called the Golgi apparatus, and you can memorize it by O and O in Golgi. So you know that the O-glycosylation happens in the Golgi apparatus. N-glycosylation happens in the endoplasmic reticulum, like everything else, like most of the changes. Okay, just the O is different. Now the third change is hydroxylation. So from the name again, hydroxylation is you add an OH group. Okay, you add the OH group to the amino acid. Now there's a disease that comes with this, because so it attaches. It attaches to proline and lysine. Okay, they can undergo hydroxylation, but the enzyme like that's needed in the endoplasmic reticulum, or like for this whole process to happen, you need vitamin C dependent hydroxylases. So hydroxylases are the enzyme that attaches the OH group to proline lysine. Okay, but this enzyme needs vitamin C. Now, since this needs vitamin C, in people who have vitamin C deficiency, you will see symptoms, and it correlates to like scurvy. That's the disease. Is that clear for everyone? Because yeah,
no, it's not.
No?
Clear.
Okay, I'll repeat it. So again, so the amino acids that can undergo, that's under hydroxylation, is proline and lysine. There are enzymes called hydroxylases. They add OH group to these amino acids, and that's why lysine can be hydroxylysine. It can be called hydroxylysine because you have OH group attached to it.
Yes.
Okay? And here
Yeah.
you have proline. When you attach OH group, hydroxyproline, proline residue, hydroxyproline. So the enzyme that that helps in this process, that adds the OH group to these amino acids, is hydroxylases. These hydroxylases, they need vitamin C. So they're vitamin C dependent. If you don't have vitamin C, they can't happen. Okay, is that clear?
Yes, okay, it's
Okay.
clear now. Thank you.
So yeah, okay. So the disease I was going into is not disease, but vitamin C deficiency can cause scurvy, which basically means like there's loss of teeth, sunken eyes, and pale skin. Why? Because the protein, like these proteins that were supposed to be hydroxylated, okay, they make something called collagen. And when there's no vitamin C, there can be no hydroxylation, and the collagen structure is not. So collagen is a protein of like it makes most of our proteins in our body, like the skin and so on. So when there's no collagen or it's weak, it leads to these symptoms. Do you get that? Did you get that part?
Yes.
Okay. Okay, I hope that's clear for everyone. Because there's one last thing. So there we talked about phosphorylation, where we add a phosphate group. We talked about hydroxylation, where you add the OH group, and this is important in vitamin C deficiency because this enzyme needs vitamin C. The other thing was glycosylation, so where you add the carbohydrate chain to it. Three things. The fourth thing that can happen to a protein after it gets translated is protein after it's being made is trimming. So there are some cases when proteins, when they get made, they're not in an active form. They're inactive, and they're called. There's something called that. Oh, sorry, I just saw the question. So there's something called Zymogens. So they're inactive. So it's when there's an increased amount of. So what do you mean increased amount of hydroxylation? I'll go into trimming a bit. Yeah, after I answered this one. So there's a decrease in. Yeah, no, there's a decrease in hydroxylation, because hydroxylation needs to happen for the collagen to be normal, like for the normal proteins, you know? So when there's no vitamin C, there's no hydroxylation, the collagen isn't as strong as it should be. It leads to scurvy. Is that clear? Clear? Okay. Should we know how hydroxylation? No, I mean, just know what I said. So hydroxylation is the addition of an OH group to proline or lysine. That's it. Is that okay? How? By the enzymes hydroxylases, which are vitamin C dependent. Is that clear?
Yes, it's clear.
Okay. And this takes place in the endoplasmic reticulum. Again, like everything else. So the only difference was the O-glycosylation that happened in Golgi. Fourth thing is trimming. So trimming is when, so like I said, not all the proteins that are made are in their active form. Okay, they can be inactive, and they can be called zymogens. So, for example, in pancreatic enzymes, they need to be inactive so that when they're made, they don't destroy the cell. These enzymes, because they're usually used to digest food, right? The pancreatic enzymes. So they need to be activated somewhere else, sometime else. So that's why they're made in an inactive form, zymogen. That's the main idea. So you can have inactive proteins made, which can be activated later on by trimming. That's it. Okay. Is that clear for everyone? Because I went through the genetic code, and then there's steps of translation and changes that can happen after the protein is made. I'm sorry I took a lot of your time, but I just have a few questions if you want to solve them. They're right here. And that's it, that's all. Okay, wait, you want me to repeat what? Which part? The trimming? And then I'll move on to questions. Okay. So let me, yeah. So in some cases, so you know how I said proteins are made? Some cases they're made, but they don't act right away. They're inactive. So they're called zymogens. Examples of this is in pancreatic enzymes or enzymes used in the stomach, for example. because they need to be active inside the stomach. They don't have to be active in the pancreas, right? Is that clear? So trimming can happen. There are like proteases that can there can be changes to these inactive proteins to make it active. So they can either cut the protein or something, and it makes it active. Is that clear? Okay. Okay. So I'll move on to questions, and you guys can answer. I'll see the chat, or you can unmute if you want to explain. So the scenario is a 4-year-old African American. So whenever they give you a clinical scenario, it's always going to sound like that. So 4-year-old African American. Okay, yeah. Someone said B already. So African American girl is brought to her physician because of episodes of extreme pain, discomfort, and then there's jaundice, and hemoglobin is low, so there's anemia. So, okay. So everyone's saying B. Can anyone tell me the disease first of all, what it is? Because I said symptoms, like you just need to know what it is. Yeah, sickle cell. Yeah,
anemia.
Yeah, that's correct. Yeah. Okay. So okay. So when I said it gets replaced, you guys are right. Okay, it's missense mutation, but the amino acid that was changed was glutamic acid. It's not glutamine. So glutamine is different from glutamic acid. That's what I was trying to tell you in the beginning. Okay, so if it's not B, what is it? Because everything else is right. Yeah, it's none of the above. So everything else here is right. It's just this amino acid, it's glutamic acid. Glutamic acid is replaced by valine at the 6th position.
Wait, so what's the answer?
So the answer is D.
Oh,
Whoever said it. Okay. So can anyone tell me A, what A is? So when phenylalanine gets deleted at 508th position, it's
Cystic fibrosis.
Yeah, cystic fibrosis. Yeah, and C is something. C is it's just wrong. Okay, good. So there's one more question. There are a few more questions, but after N-glycosylation, what is the fate of the protein? So N-glycosylation happens after the protein is made, right? It happens in the endoplasmic reticulum. So what happens after that? Does it stay in the endoplasmic reticulum? Does it get out of the cell?
Is it B?
Does the phosphate group? Wait, where? Yeah. Wait, I'm waiting for others to answer. Just tell me what you think, and I'll try to. Yeah, it's B. It's B. Because the rest of it don't like. I just put them as, they're not the right answer. So phosphate.
What's ERB?
Yeah, so ERB again, endoplasmic reticulum. That's what I meant. So it doesn't stay in it because it has to go out of the cell after it gets changed. It has to go out of the cell. And like I said, it gets into the endoplasmic reticulum so that it can be packaged and it can be secreted out of the cell to perform its function. Right? So it doesn't stay in the endoplasmic reticulum. There's nothing for it to do. A phosphate group, it doesn't need to be added. It's not necessary. And it doesn't undergo O-glycosylation because it's not a necessity. Is everyone clear with this?
Yes.
So my main point is that when a protein gets modified in the endoplasmic reticulum, its fate is to go out of the cell. And I tried to say that by saying it undergoes N-glycosylation. Because that's what happens in the endoplasmic reticulum. Okay, I hope that's clear. Next question or last one. So you have a DNA sequence, okay, and it encodes for these amino acids. Which of the following is a frameshift mutation? Okay, I forgot to put the actual sequence, but frameshift, which of these cause a frameshift mutation that causes termination of the encoded protein? So the actual sequence was supposed to be, like all of it was supposed to code for an amino acid, but one of these has a code that that has a termination codon, basically. That's what the question is asking. What do the rest think?
The answer is B.
Yeah, B. Why? Because
because there's two in the end,
Yeah.
like not
Okay.
three bases.
No, B is the right answer, but it's not because of okay, I know this is confusing, but it's not because of the two at the end. If you look at the codes, okay, so I'll explain this because it's kind of confusing. But this came in our midterm, so that's why I got it. Okay, so this is a DNA sequence, right? Because of the stop codon. Yeah, you're right. It's because of the stop codon. So if you look closely here, TCA, this is in the DNA, okay? The table at the beginning, the one with UAA, UGA, UAG, they're stop codons of the mRNA. So this is 5' to 3'. So what's the mRNA of this sequence? Like someone tell me the complementary of this.
UGA.
Yeah.
Right?
So UGA
So
is
let's
your
stop
stop.
codon.
Yeah, that's it. So she said UGA, not this way, because UGA is the 5'. I'm trying to, yeah. Wait, yeah, also the opposite. Yeah, it is the opposite. So okay, wait, I'll try explaining again.
Right?
Yeah, I want to write it down. I don't know how. So is that clear with everyone? Because the person who answered was right about, she got my point. Okay. So there's TCA here. Okay. When you have mRNA of this, opposite to this is, what is it? AGU. Now AGU is in 3' to 5'.
Can they see the
So
code?
when you flip it around, it's UGA.
We have to explain.
Okay.
Just bring your laptops.
I'm sorry. I'm trying to explain. Yeah, okay.
So this is from 5' to 3'. I just started from the second or like third amino acid. So it's GGT, TCA, okay? So this is the DNA. So in the mRNA, what's going to happen is that you're going to have it this way, 3', 5', and it's going to be the opposite, right?
Yeah.
Okay. So first of all, you're going to have to just put the complementary strands. So forget this for a second. So the way I do it, I would put the complementary strands first. So it would be C, and A.
No, it's A.
No.
AGU, right?
Okay.
Yeah.
And you just flip it.
Yeah.
Okay, so it would be UGA and ACC. So this is stuff or AG. Did I make it more confusing?
No, we get it.
Yeah. So
someone,
basically
yeah,
What
someone-
about the GC?
You mean the GC at the end here?
Yes.
No. Don't worry about that. I think there was some.
It's
Yeah.
because the
Because it's
yeah, the mRNA or the tRNA, I don't remember. tRNA is going to be reading like this, right? Once it reaches here, the whole tRNA complex is just going to disassemble. It's not even going to reach after. It's not even going to read this, you know.
So this is a frame shift.
Yeah, this is like the trick is that it's saying frame shift. Why is that? Because once you shift, for example, this before the frame shift, instead of being UGA, is UGA stop codon? Okay, so before the frame shift, instead of being UGA, for example, ACC, since it's just two amino acid, it would have been, for example, UG, and there's like something behind it, and it would be AAC. You get me? Or
No, not really.
okay, so let me explain this. Okay. Just erase all of this.
Won't the stop codon be the first? So stop codon. Yeah, exactly. Yeah. So when it's read the other way, it will be in the first place. So it won't even read the rest, you know. And the question says frame shift mutation that causes termination of the encoded protein.
What was it? UGA?
So what I want you to know from that is it causes it to stop. You need to look for a stop codon. That's all I meant.
Okay.
for the question.
Sorry, does it read till before the stop codon comes? Like, does it?
Yeah,
Yes, right?
it does.
Yeah.
Yes.
Okay.
Okay, so basically what happens is that, can you bring the camera? Zara, can you guys see or like this? Like, can you see it? The pod? Hold on.
Yes.
Okay, so frame shift mutation, that means, for example, you added 2 amino acids, okay? So let's say we remove these 2 amino acids, the ones that were added. So this would be a new amino acid. You get me?
Mm-hmm. Yes.
In the beginning, it was not a stop codon, but since you added these 2, or whatever 2 that were added, you got a new stop codon. So that's how it works. Do you get it? I think you guys are confused, aren't you?
No.
No, you're not confused, or no, you got it.
No, we
I can
didn't get it.
explain it. You get it, or sorry?
I said no, we didn't get it. Could you repeat it, please?
Okay, I'll explain it again. I'll start from the very beginning. Okay. So you have like a whole RNA sequence. Okay, so I'm just gonna write it the whole thing. CCC.
But
Okay.
can you give us the complementary for the answer?
I'm just gonna write it from the very beginning.
Okay.
CCA. CC. Okay, let's read the question again. So it encodes amino acid, okay, and there was a frame shift mutation. Okay, so frame shift mutation meaning that all of this was shifted, okay, and there was some amino acids were added, okay. Right. So let's just start from the beginning. Okay, so what do we need to do is that we need to get the complementary strand. Okay, so this would be G, G, G. This would be G, U, or A, sorry. A, U. And this would be C, C, A. And this would be A, G, U. And this is G, G. Okay, is this clear?
How is this a frame shift? Because how?
Okay. Okay, so I think the thing that is confusing you in the whole question is frame shift, right?
Yes.
Now, in the question, this is like the question I think it's missing the amino acid sequence. So in the question, for example, this is the question didn't say anything about frame shift, right? That's what's confusing you.
Yes.
Like it just mentions frame shift without giving you the sequence. Usually they would give you the sequence, but in this question, I think Momo forgot to put it, right?
Our midterm forgot to collect it, so I couldn't put that in.
Yeah.
Like it was
Well, usually they would give you the sequence.
So usually, so what I remember was for this one, there was like proline, lysine, okay, there was a specific. Yeah, I'm here, yeah. I don't know why everyone's confused. Okay, so there's a specific sequence that will be given in the exam. What they want from you is that something got changed and there was a stop codon added to the sequence. And that's why it changed the whole thing. So what they wanted from you was find something that had the stop.
Okay, so you guys get me like until this step or no?
Yeah.
Okay.
Why
But the
is
issue
this
is,
2 bases?
sorry,
Why is this 2 bases?
it's just ignore it because you literally. So what happens in frame shift mutation? You can add just 1 amino acid or 2 amino acids, so we'd have 2 extra amino acids. You get me?
Okay.
So that's literally it. And you don't need to go into details or anything.
Type, okay.
Okay. Now the confusing part comes now. Okay, so this is 5' to 3'. This one, right? Is everyone with me?
Hey, Magma.
Okay, and this is 5'. This is not 5' to 3'. This is what's confusing. You guys would just convert it, okay? You would go C, it would become with G, G with C, and so on, and you would make this. And in this, you literally have no stop codon. So this is the confusing part. So what you need to know is that to reach this transcription, or to reach this basically mRNA, okay, or the codon, you need to flip it. So some of you might ask, why do you need to flip it? Do you guys know what's a template strand or the coding strand? Okay, so this one is the template strand, okay? Meaning it's from 5' to 3'. So this one will be from 3' to 5', the opposite way. So you read it this way. This is very confusing, right? Or are
No,
you getting?
yeah, we're getting
You're
it.
getting this, or?
Yes. The
Okay.
upper one
Yeah.
is the coding.
So instead of going, for example, from GGG, you would read it GG, UGA. UGA is a stop codon, right?
Right.
Okay, so the code would stop here. Or UAG, sorry, UGA or UAG, I'm not sure. I don't remember, honestly. But I think one of these two is a stop codon. I think it's UAG.
No.
UAG, right? Yeah, so UAG is a stop codon. So the RNA, the tRNA would come here. It would read the amino acids. Okay, ignore the fact there is only two amino acids. Just ignore it, please. It doesn't really matter. Okay, so it would come here. It would read UGA. Okay, ACC. Okay, UAG. So once it reaches UAG, it knows there's a stop codon, and it would stop transcribing.
Oh, we can.
And I think UGA is also a stop codon, but I'm not sure. And I need to revise it. So I'm not sure. But my point is the very, 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 very important thing to notice. Okay, when it comes to this step, okay, you're reading the amino acid. Okay, and it's already put like this in the option. Okay, so when it's put like this, just please make sure to translate it first. Okay, so like put C with G. Okay, and do the same for the whole thing, and then flip it. The most important step is to flip it, because if you don't flip it, you're going to read this as GAU, and you won't think it's a stop codon. Does that make sense? I'm not getting any response. If you don't get it, it's okay. I have all day. So the whole frame shift mutation is not really like the main part of the question. It's just like something that happened. Okay, so you're just trying to find a stop codon. Okay, they're saying frame shift mutation that causes termination, and this is just a fancy way of saying that after you added amino acid, you have a new stop codon. Okay, so it's not something that you should be concerned with. Okay.
So for reading, we should read from 5' to 3'.
Yes, exactly. Because this one, when you match it, it would be 5' to 3', and this would be 5' to 3'. So it's going to be opposite way against each other because it's anti-parallel. Does that make sense?
Yes, thank you.
Are you sure? It's okay. Well, I can explain again and again and again. It's fine. I have all night.
No, it's clear. Thank you.
Okay, I can repeat. Okay. Okay. So which part do you want me to repeat, or from the beginning? I can repeat from the beginning. It's fine. Hello, are you there? Last part. Okay, so we started together. We said from 5. This is the code in the MCQ. The answer B, right? This code. Okay. Then the second step is which we put the complementary amino acids for it. So for C, we put G. And we did the same thing for the whole thing. However, since I said this one is the template strand, okay, the coding strand should be the opposite way around, okay? So this would be 3' to 5', okay? So we can never, okay, whenever we read something, we can never read it from 3' to 5'. It always has to be 3' to 5'. So in the exam, just to make it easy for yourself, I would take all of this and rewrite it all again. So I would write UGA, ACC, UAG, GGG. Okay, do you get it? So this is the actual final result that the tRNA will be reading. I will be reading it this way. Does that make sense? Akid, wallah, I don't mind explaining again. Because this is 100 million% is going to come in the exam. Million بالمية. I'm telling you from now, it's going to come in the exam. You just need to follow the steps. Read the MCQ or the MCQ, okay? Translate it, then flip it. So reading, translating, flipping, okay? Makes sense? If it doesn't make sense for one person, I'm going to repeat it again. I don't mind.
Okay, it makes sense, but in this question, the one in the slide.
Tell me.
What should we do, Yani?
What do you mean the
Look
question?
for the frame shift?
No. So the one in the question, what you do is you go through the MCQs. Okay, you go through the option A, for example. You go through option A and you do the same thing. You get me? And you go through option B and you do the same thing. If you find the answer, khas, no need to do for the rest because you find your stop codon, which is UGA or UAG. But if you don't find it in, for example, option A, you go to option B and you do the same thing. Then you go to option C and you do the same thing. So just in case, have a pencil and paper. They're going to give you a paper. Just have a pencil with you and the exam to write this stuff. Okay, it's not complicated. Yani it's going to take maybe 2-3 minutes, but yani, this is a free guaranteed answer. Yani. Do you get it? If you want me, I can do it for the other options, just to make sure that you get it.
No, I understand. Thank you.
Everyone gets it? Tamam, alhamdulillah. Yay. Anything else you guys don't get in the lecture?
You guys get everything. I'm sorry if I confused everyone with this question, but if you got it now, that's good. Did you get it?
Yes, thank you.
Please, if you don't get it, this is literally gonna come in the exam, 100%. If you don't get it, don't be shy. I
want to say,
and I won't be sad, and I won't be tired or anything. I literally got nothing until the end of the day. I can explain it. I can explain it for the other options if you want.
I can explain.
We can pull up another question if you want, and it's up to you.
Mm-hmm.
Do you guys get it? I can search for another question if you want. You guys want to try?
Yes, please.
Okay.
Okay. I'll search for another question.
I just want to say something while he searches. So we got this in the midterm. I remember seeing GC and I put this answer. I remember Anush said, I think it's because of that. But then I realized later on that it's because of the stop codon that was here. So don't make that mistake. That's what I'm trying to tell you. Okay.
But there's not stop codon.
If there's no stop codon,
yes,
that's not the answer. There will be one that has a stop codon. This frame shift mutation is confusing you because, like I said, frame shift mutation can cause there to be there can be a termination codon because of that, and that's what the question wants. That's what the question is asking for.
Rewrite one for them.
Okay.
And
thank you.
Is everything in the lecture clear before I go to the example for you? So everything before this clear, the changes that happens, and then there's okay. Is that okay? Okay, we'll move on to one more example so that you guys can practice on this one. Okay, that's good.
You want to Google it? Tell them I'm gonna write the question. Like, just give me 3 minutes.
Yeah, just 3 minutes.
Whoever doesn't have any question, or
whoever doesn't have a question, you can leave. It's fine because I took so much of your time. I'm sorry. I hope preparations are going well for the midterm. I hope this helped too.
Three codes. These 3 codes are DNA codes, okay? And we want to make them, we want to know which one have a stop code, okay? So remember the 3 steps. So first of all, translate, flip, translate, flip, then read it. Yeah.
Okay.
Also, don't forget, you're making an mRNA. So means if you have an A, you write a U. You don't write a T, okay? Because you might write a T and miss a stop codon. All the stop codons have a U in them. So. Yeah. Which answer? Put A, B, C next to them so we know.
Yeah. Wait, let me.
Oh, that's like another part of the code.
Sorry. Wait. A.
Okay.
Everyone saying
A. Rima's iPad. Okay. I told you there's more than one option, so it might be B or C as well. So you might want to go through.
Everyone's saying A. Why isn't it 3?
It is 3 as well. Yeah. Oh, someone paid attention.
GGG, yeah.
See, here you go. You're translating it. You're doing very well. Now you're getting the point. How to
Yeah.
do it.
That's the right way.
But my only point, no, actually, it's very great. MashaAllah, everything. You got every code right. ANC,
ANC.
of course. Momo sister got it.
Good job.
ANC.
ANC, MashaAllah. You guys
Everyone's
are doing
getting
great.
it.
Oh, now,
You guys
now
are-
I can
guys-
sleep knowing that every single one of you guys who stayed here will get
Yeah.
this question right. MashaAllah. Great. Anything else we could help with? Anything you didn't get in the lecture? It's your chance. We're gonna help you with anything. I'm literally
Yeah.
ready to explain any concept.
Is everything good? Clear?
That's the writing.
Yeah.
Question.
Okay.
Thank you so much, you guys. Good job.
Thank you.